Welcome to Tell Us What You Know, a series from Drug Discovery News where we ask scientists to break down some of the most pressing topics in research today. In today's episode, we're exploring the growing field of spatial proteomics, a powerful technology that maps protein expression within tissues and cells to reveal new layers of biological insight. We're joined by Junqi Liao from Sincell, who will help us unpack what sets spatial proteomics apart, what challenges this field is facing, and how this approach is opening new doors for drug discovery. Let's dive in. Hello, JC. How are you?
Good. How are you, Yuning?
I'm good. Thanks for joining us today. So, first of all, can you tell us what is spatial proteomics and why is it gaining so much attention in biomedical research today?
Yes. So, spatial proteomics, as you know, recently getting a lot of interest. Uh, it's aiming to understand the protein contents in terms of location so that one will know specific proteins where they are in terms of disease-related spot or understand the relative location of different protein in a large scale. Um, as we know, 95% of the drug is targeting proteins instead of RNAs. So protein pretty much is a primary functional molecules in cells, but they are not always easy to know where locations. If you know the RNA, you probably still don't know the location in proteins in terms of the inside the cells. So it's really important that we understand the kind of a protein locations in this whole disease samples or some biological samples, so you have a better understanding of the functions and then hopefully the downstream drug discovery.
What do you think is the biggest challenge facing the spatial proteomics field right now?
Yeah, in Nature Methods, in the 2024 Methods of the Year, they chose the spatial proteomics as a method of the year. And then as in this article is basically there are two categories of spatial proteomics. One is targeted method, and the other is unbiased method. So all the targeted methods are antibody based. So the major challenge is one cannot find novel protein contents beyond what we are already know. For example, we are still calling negative breast cancer triple negative because we still have a hard time to kind of categorize what we saw in different samples of triple negative breast cancer and then identify really the positive biomarkers for that. Um, there are other things. And then for the existing unbiased approach, that's pretty much based on laser microdissection. The challenge is in the location precision. The laser beam is pretty big, so you will have a hard time to isolate, for example, in the specific, like the Lewy body of Parkinson disease is too small. And then, so I would say the biggest challenge is in the spatial proteomic discovery for disease-associated locations.
And how are you addressing that challenge in your own work?
In Sincell, we call our technology microscoop. Scoop is like ice cream scoop. Is basically pretty much like a metaphor of isolating the protein in the location you are interested in. When I developed this technology, I basically just want to solve my own problem in biology. I want to study what proteins in the primary cilia of different kind of contents. And so our technology basically is just using laser light to trigger protein biotinylation. So labeling the protein in the location you're interested in by light. And because we are using light, the precision is very high, is in the order of 100 nanometer. So we can basically have using light to tag the protein in the location you're interested in. And later on, we take enough, so we use automatic, basically all the electrical control to shine light to many of the, for example, beta amyloids of Alzheimer's or the, like what I said, Lewy body of Parkinson's. Then you can take enough protein and then later on you use biochemistry to pull this protein out. It will become very pure. And then this is like I would say this is probably the first high precision spatial purification method. And with this purification ability, now you can do high precision spatial proteomics. And with the downstream mass spec, now it becomes possible for discovery of novel protein constituents in the disease locations, for example, in the aggregates of Parkinson's diseases, in the nuclear condensates of triple-negative breast cancer, or even in the location of ADC drugs. So this all become possible. So this is kind of our technology trying to help, and then hopefully the protein you find in a location will lead to the future further biological studies so that we can find the future first-in-class protein drug targets for many diseases.
Can you share an example of how your spatial proteomics technology led to a discovery that opened up new directions for drug development?
Yes. So as a synthetic microscope is still in a pretty early stage, but we already see promising outcome, not directly already finding the new drugs target, but leading to that direction. One example is Dr. Christiana Longu from University of Stuttgart recently published a manuscript in bioRxiv showing that she used our microscope technology and identified novel protein contents in the nuclear condensate of triple-negative breast cancer. So when they get rid of this one of the protein they found by knockdown, they show that the cell motility of these breast cancer cells is reduced. So it's possible that when it may be a potential future drug target, right? You push it down by drugs, then you can kind of suppress the metastasis of triple-negative breast cancer. That's the hope. I mean, if we do more and more of these kind of studies, then this high precision spatial proteomics will become an engine to generate therapeutic hypotheses for first-in-class drug targets. And the other project that internally we have run is the interface between the mitochondria and lipid droplet for fatty liver studies. So we can see some new protein contents in the contact site between the mitochondria and lipid droplets. So, and then we get rid of one of the protein. We show that the mitochondria change the shape, the contact between the oil and the mitochondria reduce. So it is possible we can kind of disrupt the oil effect to our metabolism. Again, this is still very early study. I just want to say that with these two examples, it's possible we can have more and more with downstream biological studies and then broader kind of usage of our technology for different diseases.
Finally, what's something unexpected or exciting you have discovered using your spatial proteomics technology?
Yeah, so as I just said, I feel that our finding, as we know, pathologists, they are very good in like seeing the microscopy image, and they immediately know, okay, what disease this is. They can recognize hundreds or more than hundreds of images, like different kind of diseases. And our job is to not only help them identify what disease is, but when they see the spatial feature, we can isolate the part that's weird. For example, we have done that with one project is we work with Mayo Clinic. They send human brain sample from ALS patient, and we isolate the protein in the TDP43 aggregates, and we identify protein contents. So I would say this kind of thing is the most exciting that to help the doctors or pharmaceutical companies to find the protein contents in the special diseases. And I mean, we have done some biological studies. For example, we isolate the protein from stress granules. And the most exciting thing is that we bought more than 10 antibodies, and almost all of them show positive in the location we are interested in. And all these 10 has never been mentioned in any literature, so related to stress granule. So I think this is the kind of the excitement. I mean, when I did that, I didn't expect we get such a good specificity that as long as you isolate this location, we can get pretty pure proteins and then tell people what's in that location. So I guess this will be what I'm most excited about. That hopefully this can be used for a protein related to Alzheimer's, triple negative, fatty liver, or or basic science. For example, how our memory is stored in our dendritic spine when mouse or some animal model learn something. So I think this is what I see this technology, and I think the excitement with the spatial proteomic discovery capability.
That's really good to know. Well, thanks again for your time with us, JC.
Okay, thank you very much, Yuning.
If you enjoyed this episode, be sure to like, subscribe, and check out our other videos for more expert insights on science. Thanks for watching. We'll see you next time.